Very good morning. Today we are going to discuss about seed production in Castor. So as far as that Castor is concerned, these are both variety and hybrids are available. So regarding that seed structure of the Castor, so this is having a leafy cotyledon. Already we discussed in sunflower seed structures. The sunflower also have a leafy cotyledon. So similarly in the castor also have a leafy cotyledon. So apart from leafy cotyledon, on outer side one provisions are available. This is the one. So this is the one is normally known as a caruncle. So this caruncle play a major role with reference to that germination related process, germination related event occurrence in the castor. Similar kind of caruncle structure is available in the case of rubber as well as that tapioca seeds also. So this and all externally arranged such as. Apart from leafy cotyledon, these have a well-developed endosperms also. Even though this is a dicot, so this put it under the albuminous category. So normally the castor seed, that outer structures that will have a brown color, that shell kind of things. So this is the one, the brown color shell. On inner side of that shell, one more white color layer will be there. That layer completely surround that endosperms. You can split open that endosperm into two half. You can split open into two half. If you split open that endosperm into two half, or inner side, the middle part, you can observe this kind of a leafy cotyledon. So the leafy cotyledon are white in color, not a green color, it's a white in color. That will have a vein like a leaf. So whatever you are seeing here, the similar kind of vein arrangement, everything is available. So this also comes under that leafy cotyledon category, like that of the sunflower. So this is a structure of that castor. So this one is that external structures. So external seed coat may be a brown in color or white, or some different patches will be there. Regarding that floral biology of that castor is concerned, so this will produce both male and female flower separately, but both of them are going to be occur on the same spike. So that raceme is produced. So both one on the same spike, and that the top portion is occupied with the female flowers. Female flowers means that is here whatever you are seeing in a red color, that is a female flowers. So that will occupy the top. So that occupy 40 to 60% of the area. The top area is occupied by the that female flowers. The bottom, mostly that 1/3 of the spike, the bottom portion is allotted for the female, that male flowers. So here that male and female flowers are produced separately, but both of them going to be there one on the same spike. So because of this is highly cross-pollinated. So the main pollinating agents are wind, birds and insects, and the inflorescence mainly produce at a terminal part. So the inflorescence occurrence in a terminal part. This is produced in a terminal part, and that inflorescence is a protokinous nature. So like that of that pajra, so this also a protokinous nature. So the gynoecium that attain mature first before attainment of that pollen maturity. So that is a thing. So this also favor for occurrence of the cross pollinations. So I already told you. So that female flowers are seen on the top 30 to 50%. Here you can see the red color one, whatever you are seeing, that and all the female flowers. And the bottom area alone is allotted for that male flowers, that is a yellow in color. That anthesis normally occur between 8 a.m. to 12 noon, and female flowers only we can expect that seeds. So the proportion of that female and male flower that are going to decide the success of the cross pollinations. So that is a important thing as far as that castor is concerned. So that these have different sex forms like monoecious, pistillate, interspecies staminate flowers, and sex divert, and like that 4 different sex forms are available. If it is a monoecious, the same spike, the 1/3 to half that the bottom will be occupied by the male flowers, and the remaining top area will be allotted for the female flowers. That is a monoecious nature. That another kind is a pistillate nature. In the case of pistillate spike, so that the entire area is covered by the only female flowers. So 70 to 99% of the area is occupied by the female flowers alone. Very few number of male flowers alone will occur. So that too in the basal. So if at all these kind of things are there, that is a pistillate flowers. The third category is the interspaced staminate flowers, in short called as ISF. So it's a variant of the pistillate form. So here majority of them are female flowers, that is spike will have majority of them female flowers, but here and there throughout the spike, here and there that male flowers are occur. Okay, that is the interspaced staminate flowers. Another category is a sex revertant. So sex revertants are originally it's a pistillate flowers. Originally it's a pistillate flower, but at any point of time that will be reverted back to monoecious nature. Monoecious means that both chances are there for occurrence of the male flowers also. Both male and female flowers will occur at any time. So that reversion is under the control of the climatic, soil and environmental factors. So for occurrence of more number of the female flowers, the following conditions should be prevail. If you cultivate that crop during that winter season, coupled with low temperature of less than 30 degrees Celsius, and the flowers that appear on the early age of the plant growth, that is the early order spikes, and a high level of soil nutrition, and less difference between the day and night temperature, highly favorable for female flower production. So this is otherwise called as female flower production promoting environmental conditions. Similarly, for that male promoting environments are summer season, rainy season, high temperature, that is temperature should be more than 32 degrees Celsius, and the age of the plant should be old, and a late order spike, and low level of soil nutrition, and large difference between day and night temperatures that promote that male. So these are all the reason for occurrence of the sex revertants. Sex revertants are originally a pistillate. At any point of time that may be reverted back to the monoecious nature. Here you can see that images for all those category. So this is the first diagram. This is an pistillate flowers. So here you can see only that female flowers alone. That entire spike is occupied with the female flowers only. So red color one. Whereas that the second one, this one, interspaced staminate flowers. So I mean it's a raceme with only that male flowers, because here whatever the flowers are there, everything is yellow in color. So that yellow is a anther one. So here that everything is yellow in color and male flowers alone available. So this is a staminate flowers, whereas that C, that is an interspaced one. Originally it's a pistillate, but here and there some male flowers also will occur. That staminate flowers also occur. So because of that, this is a ISF category. The fourth one, that is that equal proportions. So this and also output of that female flowers, whereas this and all staminate flowers, male flowers. So both male and female occur in equal proportions. So this and all images regarding to that different sex forms. So this is a sex revertant. Actually it's originally it's pistillate nature. Due to that environmental all those influences, at any point of time this will be reverted back to that monoecious nature. So here you can see that yellow color staminate flowers started to appear in this. Here also you can see, so this is pistillate flowers. So only that female thing alone will occur. So everything is a red in color, whereas this is an interspaced one. So interspaced one, here you can see that occurrence of that male flowers here and there. So that is interspaced one. So this is a monoecious one. Here that both the things are there, one of the same spike. So this and all the male, so this and all the female flowers. Whereas here this is a staminate flowers, because here that predominantly the male flower is produced. So this and all images corresponding to that various sex forms in the castor. So now in the case of castor, we have a different kind of pistillate mechanism. So the pistillate mechanism is very well exploited for the purpose of hybrid seed production in castor. So 3 different pesticides mechanisms are available. The mechanism like N type, S type, NES type. We can discuss one by one. So first we are going to discuss about that N-type mechanism, or otherwise known as a conventional mechanism. So that N-type mechanism is almost almost equal to that of your GMS, genetic male sterility system. So that genetic male sterility systems we are using hybrid seed production in the case of red gram. So the exactly same one is here call it as a N-type mechanism. Here also that sterility related things and all governed by the recessive sex switching genes, and this also need to be maintained through symptomatic, that is crossing with the close relatives only. So here also the same problem is there during that multiplication of that female parental line. That 1:1 segregation will be there in order to ensure the plant with the complete pistillate class. We have to go for that roguing. So we have to rogue out that plants. 50% of the plant we got rogue out. So the same thing is there. So that is N-type mechanisms. So here the problem is there. So uneven emergence and variations in time of the flowering, high percentage of the monaces, all those things will be expected. But here that expected thing is in the female rows, whatever that plants are there, that should be expected to produce only the pistillate class. But here chances are there for occurrence of the monaces, so we got to do that roguing. So whatever the problem we are facing under GMS systems with reference to female parental line multiplication and maintenance of the female parental line material during that hybrid seed production, same problems also prevail here. So this is somewhat problematic one. So because of that now this is not gaining that much importance with reference to hybrid seed production castor. That another mechanism is S-type mechanism, or otherwise known as a non-conventional mechanisms. It is derived from the reversal. So normally this start out as a female. So this start out as a female, started to produce only that pistillate class. But at any point of time this will be reverted back to that monaceous. At any point of time it will be reverted back to that that monaceous. So this also highly problematic one because of that stability is not there. Stability in expression of that pistillate mechanism is not there. So because of that this system also not that much useful for hybrid seed production castor. So now we are adapting that another system, that is NES systems. So this line is normally pistillate. So these particular plants are produce pistillate class only under moderate temperature. So the temperature is moderate and favorable temperature there, that produce only that pistillate class. But all any abnormal temperature prevails, at the time only there is a chance for occurrence of the male flowers and an interspaced manner here and there, not like that of your monaceous nature. So because of this is somewhat better system when compared to that previous two systems. The only thing what we have to do is during that hybrid seed production operations, if at all any interspaced things are going to be there, through that manual roguing we can ensure that 100% pistillate things in the female parental rows. Another thing is based on that temperature prevalence, during that moderate temperature prevailing conditions we can go for that hybrid seed productions. Whenever that temperature go beyond that favorable temperatures, at the time we can go for that that parental line multiplication. So this is a way in which we can very well utilize this mechanism for hybrid seed production and hybrid development in the case of castor. So now this system is very well utilized. So now we have our indigenous line VP1 also, pistillate line is available. So this line is very much used for that hybrid development program. So the Gujarat, they are the leaders with reference to that castor research is concerned. From the Gujarat only they released the first castor hybrid as far as that India is concerned. But USA they are doing a very wonderful work with reference to that hybrid development and hybrid seed production in castor. So in India that Gujarat is the leading state, followed by Andhra Pradesh with reference to that castor production. With reference to plant anatomy, in the case of castor, that have the unique mechanism of the blooming. So occurrence of that white waxy layer, here you can see white color waxy layer that will appear on the leaf of both the undersurfaces and upper surfaces and stem and pedicel and all those things. But some other variety that may be appear in under surface of the leaf, some other variety that will be in the upper surface of the leaf. Based on the occurrence of that bloom, the varieties are divided into different category. No bloom category. So here that bloom that won't occur in any part of the stem or leaf or any plants. Single bloom, so the blooming will occur only on the stem. So only on the stem. Double bloom, the bloom will be on the stem, petioles and lower side of the leaves. Triple bloom means the bloom, the white waxy layer that will appear on all parts of the above ground. So that is a triple bloom. The advantage of occurrence of that bloom, that is a white waxy coating, is that gives some sort of natural protections to the plant systems against extreme weather condition like cold, drought. Apart from that, whatever the plant or whatever that castor variety or hybrid that are having that blooming mechanism, that shows some sort of resistance against the acids. It's a serious pest as far as the castor is concerned. So that how the resistant against that acids. So that is the thing. So this is the plant anatomy is concerned. Regarding that seed production, so this is considered to be a cross-pollinated one. So because of that, here this given is wrongly, but here we have to maintain 600 meter isolation distance for that both variety and hybrid at foundation stages, and 300 meter for certified stages. So 600 meter isolation distance both for variety and hybrid during foundation, and 300 meter during the certified stages. Regarding that season, the Kharif season normally ideal for male promoting environment. During that times we can go for that varietal multiplications, male and female parental in multiplication of the multiplications. Rabi is a right time for hybrid seed production. So Rabi is recommended for hybrid seed production. So that seed rate is for varietal seed production 10 kg per hectare. For hybrids we have to use 4 kg per hectare as a male parent. A female parental line seedling material 6 kg per hectare. And spacing also long duration 19 to 90 and short duration 16 to 30 centimeter we have to follow. For hybrid seed productions we have to follow the planting ratio of either 3:1 or 4:6:1. So normally that Gujarat Castor hybrid 4, that there is a problem of that synchronization. So there we have to follow that staggered sowing. So the male parent should take up sowing 7 to 10 days. That the male takes 7 to 10 more days for flowering. So delayed flowering will be there. So in that case male plant should be planted early. Manures and fertilizers like that of that agriculture crops. Here also we have to apply 12.5 tons per hectare farm manure and 15 kg of NPK per hectare. So irrigation also very critical. So during primordial initiation, flowering and pod development, we have to ensure that water availability. Rogging also very important operation because it's a cross-pollinated crops. So to maintain that prescribed level of the genetic purity, so we have to do that rogging. The different stages like 10 days prior to flowering, during flowering, a week before first picking, after first picking. So all those stages based on the given characters, we have to look for presence or absence of the off-type populations. So if at all any off-type populations are there, that should be removed from our field in order to ensure that desirable level of genetic purity in the resultant seed material. Harvesting, so here that as and when that the capsules, the fruits are called as a capsule. The immature fruits are green in color. As and when they attain maturity, that will become brown in color. So based on that browning symptoms, we have to identify. Another main thing with reference to that castor is concerned, that spike and all produced in a particular certain days of interval. Say example, the first spike appear today means another 15 days only that next spike will appear. So sequential spiking normally occur. So that will be favor for harvesting of the capsules at different times. So we can follow that 3 to 4 pickings we have to do. So first picking will start from 90 to 120 days. After that, once in 25 days we have to go for that second and third and fourth that harvestings. So dry the capsules with the sunlight by keeping in the shade. Then after that, with the help of the shellers, we can separate the seeds from the capsules. At any cost, we should not harvest that premature capsules. If it is a premature one, definitely that will cause us problem with reference to that seed viability, and oil content also will be very less. So these are the things with reference to that castor harvesting is concerned. And then these images corresponding to various stages of that seed development. So here you can see that days after, and this is during that 61 days after pollination only that will attain that almost that matured form. So before that, this is in the process of the development. So this is a inner view of the cross section of the capsules. So these are capsules inner views. So for grading purpose, we can use at 8 by 64 inches round perforated metal sieves. For storage purpose, we have to treat the seeds with carbendazim 2.5 gram per kg of seeds. We can store the seeds for longer periods. So as far as that seed standard is concerned, so here that minimum germination percentage per seed is concerned, 70% we have to maintain. The pure seed percentage also 98%. And yield wise, if it is irrigated crops, 2000 to 3000 kg per hectare we can harvest. If it is a rain-fed crops, there we can harvest somewhere around 1000 to 1200 kg per hectare. So with that we are completing that castor. The next one we can see that sesame in the next videos.